I'm going to start my timing duties and turn it over to our first presenter, Dr. Michael Opata. Michael, you can go ahead and share your screen and get started.
Can people see my screen? Good. Okay, thank you, Kate, for that for the invitation to give a talk on my research. So this research was funded by the URC grant, and it involved some students. So I had 2 graduate students and an undergraduate student. The research was to look at how moderate malnutrition affect the immune response to malaria and also alter gut microbiome or gut structure. So to do this, we had 3 groups of mice. So these groups were fed a controlled diet or a moderate malnourished diet. So we call this a moderate malnourished diet because it has all the calories that are required, the same as the control group, but it is deficient in zinc and iron, and it also has 3% proteins instead of 17% proteins for the control. So let me get my pointer. So what we do is we feed them for 4 weeks to induce the malnutrition. Once we have induced malnutrition for 4 weeks, we either infect or don't infect. So we infect one of the malnourished group and one of the control group, but the other groups are not infected, but they get the malnutrition or the diet. Then 9 days later, which is the peak of infection, we either feed the mice on dextran just 4 hours before we sacrifice. This dextran is a molecule that is labeled with FITC, so it is fluorescent, and it is a big molecule. It's not supposed to pass through the gut. So when we feed and there is damage in the gut, then it will penetrate through the epithelial cells and enter the blood. So we are able to detect it in the blood. So we wait for 4 hours and then we sacrifice the mice. We collect the blood so that we can check if the dextran leaked into the blood. So it will be a measure of our gut leakage. And then we also collect the mucosal tissues, the small intestines, large intestines, and cecum, take pictures, measure the length, and then do microscopy and also analyze immunity. So what we saw is that in the blood, when we take out, when we measure the dextran, looking at the uninfected mice here, the green and orange, we see that the malnourished mice that have been given dextran have an increase in dextran in the blood as opposed to the control. But when you look at the infected, the blue control mice, they also have a higher amount of dextran, suggesting that malaria infection alone induces some gut leakage. When we look at the malnourished mice, that leakage is further increased, suggesting that both malaria and malnutrition increase that leakage even more. So when we take the pictures looking at the infected and infected groups, which are the control, we have highlighted in green, we see that there is no difference in how they look, both the control and the malnourished. But after infection, we see that the malnourished mice that are infected, the small intestine look yellowish as opposed to the control diet. So suggesting that the malnutrition together with the malaria infection is doing something to the gut. When we measure the length, we see that the small intestines in the malnourished mice that are infected are much shorter. So the red, suggesting that there is something happening to these mice. So what we did is we sectioned these small intestines and then do a H&E staining to determine if there is any damage. So the top panels are the infected or uninfected, and then the bottom panels are the infected. On the left is the control, on the right is the malnourished. When we look at the uninfected top panels, we see that the epithelia is good. So suggesting that there is no damage. But upon malnutrition, even before infection, we see that there is disintegration happening of the mucosal surfaces. After infection, we see there is a little bit damage, but it's not much. But in the malnourished mice, the damage is even much higher. So indicating that malnutrition together with malaria infection is causing high damage in the mucosal surfaces. So we took these tissues, both the small intestines and large intestines, and then mashed them to determine immune response. So in our gut, we have normal flora, which is composed of bacteria. What we do is inside the gut, there are immune cells that are supposed to protect or sample any pathogenic bacteria that we might ingest. So we can determine those immune cells by looking at CD11b, which measures macrophages, or CD11c, which measures dendritic cells. These are just innate immune cells that sample the gut. When we look at the uninfected, so the green and the orange, we see that there is no effect on these immune cells when the mice are not infected. In both all the immune cells that we are looking at. But upon infection, red and blue, we see that the infected malnourished mice, which are red, have a decrease in these immune cells, both in the small intestines at the top and large intestines at the bottom. So indicating that malnutrition together with malaria infection is causing mucosal damage and altering the immune response in the gut that is supposed to be sampling. So with that, I will end there and welcome any questions.
Thank you, Michael. We have about 5 or 6 minutes for questions for Michael. You can unmute or you can type them in the chat.
That was really fascinating. And I know I've followed Michael's work previously, but it makes me want to say, like, what's next? What's your next step? And I know you've got some things going on, but maybe just like the next most recent step.
So that is a good question. So because I will go back. So, sorry. So based on what we have seen, that these epithelial cells are damaged. These epithelial cells are composed of tight junction proteins. So we are interested in isolating the protein, the cells from the small intestines and large intestines. And then we want to look at the damage that is caused on those tight junction proteins. So we have different tight junction proteins like occludin and claudin, and ZO1 and ZO3, which are important in holding the mucosal surface close. So we want to see if that is being damaged.
Okay, thanks.
You're welcome.
It's very exciting.
And Michael, can you zoom out for those of us who are not biologists and tell us why it is important that you are showing that malaria paired with malnutrition is so damaging to the guts? Why is this important for us to know?
So it is important for us to know because the main thing is mostly malnutrition is something that happens to most people. Sometimes it happens because of poverty or lack of good nutrients, but that is a problem for developing nations. For developed nations like the US, malnutrition happens because of malabsorption. So you are eating the right nutrients, but there is a defect in your body absorbing those nutrients. So if your body if you have a defect in your body absorbing those nutrients, then you'll be malnourished, moderately malnourished, because you'll not get those nutrients. So any immune response happens the same to pathogens. We are using malaria as an infection model for African regions where malaria is endemic and also malnutrition is endemic, but this can apply to any developed nation that there is a problem with malnutrition. So one of the other things that leads to malnutrition in developed nations is food insecurity. whereby rural areas don't have access to food the same as towns. So that can also result to malnutrition since they can only have access to foods that are in their environments.
That's super relevant and important. Thank you for clarifying for the rest of us. Any other questions for Michael?
I have a question. Michael, thank you so much for this presentation. This is really fascinating. I'm going to kind of zoom out and just ask you more of a broader question on your opinion on some things. I have personally had some gut issues and dealt with what ended up being a medical disability and saw a number of doctors. And so through that process, I learned a lot about some of the conflict around this idea of leaky gut, and how some of the research and Western medical practitioners were sort of at odds with more Eastern medicine or alternative medicine, whatever sort of category you want to label them with. And so given your presentation today, I'm sort of leaning toward, okay, well, it sounds like leaky gut is kind of a real thing, but I wanted to get your opinion and just have you respond to that piece, what happens in medical practice. And is leaky gut real?
Yeah, so for the part of leaky gut, yes, it is real. And that is dependent on different things. So for patients who are suffering from Crohn's disease or IBD, then there is inflammation in the gut. So this inflammation is because of uncontrolled immunity happening in the gut, or some of the normal flora that we have in our gut misbehaving or outcompeting for nutrients. So when they outcompete for nutrients, some of the microbiota that is supposed to be normal and protect us becomes more pathogenic. So it causes damage. So that damage can lead to leaky gut, whereby some of the microbiota are now able to come out of the gut and then go to other tissues like the liver or kidney and cause other peripheral organ failure, which is a big problem. And the problem with most of the medications that are given to people who are suffering from Crohn's disease or colitis, they are anti-inflammatory. So they are not specific to the gut alone. They will reduce immune response to the body in general. So it makes those people more immunocompromised in case there is another opportunistic infection that can take advantage of the anti-inflammation that they are being given.
Thank you so much for that.
Yeah, super interesting, Michael. Thank you so much for your time and your presentation, and congratulations on your funded project. And we wish you many future successes, both internally and externally. Quinn, we'll go to you next.
Thank you very much.
Quinn, you're muted.
Yep, got it. Sorry. Is that showing up? My screen
Yes,
showing up?
yes,
Okay.
we see it.
All right, great. Well, thank you again to the Office of Research for hosting this. So I want to talk a little bit today about network dynamics, and another topic that we all have heard more about than we probably wish in the past 18 months, which is COVID-19. So before we get too far, I'll mention that this is collaborative work with Renee Salinas, Lynn Sieferman, and Brian Burke. And this was all supported by a COVID-19 multidisciplinary research cluster grant. And so this was really work done over last academic year, 2020, 2021, but is ongoing. So when we think about mathematically modeling a disease outbreak, I think of this as sort of happening on a spectrum. And that spectrum depends on sort of how much information we have, both about the disease, but also about the people who are being affected by the disease. So on one end, we have very basic, what we call compartment models, where every person is just assumed to be susceptible, infected, or recovered from the disease. So this is so-called SIR model. But you make a lot of assumptions here, like there's homogeneous mixing of the population. So, everybody's interacting with everybody. You have to make lots of unrealistic assumptions rather, to be able to make the model tractable to do any sort of analysis. On the other end of the spectrum, you could try to just gather a bunch of data. So if you knew exactly who every person interacted with on a given day, right, you could build an exact contact network, and then you could simulate potential transmissions on that contact network. And there's a whole host of sort of gray area in between these two ends of the spectrum, where a lot of research goes on. But sort of unsurprisingly, maybe, universities have lots of information about how students interact with one another. So just as sort of a few examples, we know their course enrollments. We know where, at least if they're on campus, we know who their roommates are. We know every time they swipe their app card somewhere. We know what student groups they might be affiliated with, or at least some of them. So we have a lot of information actually on how students interact with one another, which makes building a contact network a pretty realistic option in this scenario. So what we did is we started by building, we didn't use all of this data, but we started by building what I'm going to call the co-enrollment network. So essentially in the graph, in the picture that I'm about to show you, there are a bunch of dots, like almost 20,000 of them. And 2 dots are connected by a line. So each dot represents a student, and 2 dots are connected by a line if they have a class together or they both live on campus and they're roommates. Okay, so here's a visualization of the university. So you'll notice that there's sort of a lot of distinct sort of clusters on this picture right now. The largest component that you see down here in the bottom left consists of about 16,300 some students. That's most of your undergrad and grad student. I mean, that's most of the students at the university. And then these other groups you see, they're actually sort of very specific programs. So there were 4 students in the Master's in Athletic Training program. They only take classes with each other. None of them lived on campus. So their only contacts, at least at school, and by contacts, I'm thinking sort of, that I can easily quantify, are with the 4 other people that are in all their classes together. Okay, and we see similar things in other graduate programs, and also the seniors in the BS program in nursing. But okay, these are sort of the outliers. So we're really going to focus on this main cluster here. I'm going to skip over those sort of facts. So let's look at this sort of main cluster here. It's about 16,300 students. Immediately, just by generating this plot, we get some information. So I've actually coded here, everything is either red, orange, green, blue, or purple, and the blue and purple are a little hard to see. But you can see clustered here in the middle are all these red dots. Those are actually all the first-year students. Orange are sophomores, green are juniors. And what you can already see is that freshman students mix with a lot more students than, say, a graduate student. Like these students out here are graduate students. Okay, so already we start to understand something about disease transmission and issues we might have just by looking at this picture. As a quick example here, so this is just that same graph, but with all the dots uncolored and the lines removed. So you can maybe see here, I've colored one of these dots in sort of the center red. It might be sort of hard to see, but there's one red dot there. So imagine that's one infected person. In green, I've now put all the people that red person comes into contact with in a given week. This is just through housing and course enrollment. Then I look at all the people. So let's say they infect all the people they come in contact with. Now these are all the people that all the green people are in contact with. Okay, now we're up to almost half of the student body, not quite. And then if I look at all the people that those orange people come in contact with, well, it's hard to figure out if anybody isn't potentially infected anymore. So this is actually a property that you can study mathematically. It's called the small world phenomena. So if you take any 2 students and one of them is infected, the likelihood that they infect another given student is very low, because it's unlikely that I'm in another class with any given student. However, what is very likely is that we have a classmate in common. And what's even more likely is that we each have a classmate and those classmates are in a class together. So sort of the transmission chains between any 2 students are fairly short even though one individual may not be connected to a lot of people. So once we have this, we can, as sort of I've shown you here, we can simulate disease outbreaks. Now, I'm not going to infect every person if I'm contagious. I'm not going to infect every person I come into contact with. But using some reasonable parameters, we can sort of run a bunch of simulations. And so that's what we did. So we sort of ran these over a range of scenarios based on different parameter values, which I won't get into for the sake of brevity. And so first, we were doing this in fall 2020. So we said, well, what if things were really good? What if it's not so contagious? Students are really, really diligent about not partying and monitoring their symptoms and wearing their masks. Well, this showed us something that we now know absolutely didn't happen, which is some students will show up infected. They'll all be so nicely behaved that the disease will die out. And, by October, we'll be great. Okay, well, that didn't happen. All right. We then looked at some sort of mid-range scenarios. And so these mid-range scenarios were, okay, maybe it's a little more infectious and students aren't quite as good about not partying, but they still try to be good and okay, things aren't so bad. And in this case, what we saw is sort of an initial spike in infections. And then we get what I sort of called the slow burn scenario, where we sort of have these localized outbreaks. So really what's happening here is students are, they're going out and partying and it gets another spike and it sort of dies down. And then there's another party. So we get this sort of slow burn scenario. We actually sort of know that sort of happened, but we know there was a major peak. And in fact, when we looked at sort of our worst case scenario, that's exactly what we got, is sort of this noisy peak. And so here you have a peak. Let's see, I think I have it over here. Right? You have a peak occurring about 90 days in. And you have about 3,000 to 4,000 new cases being generated from community transmission here. If you actually look at this peak here, this is actually measuring the total number of symptomatic cases. And I think this prediction was that at the peak, we would have about 200 symptomatic cases. Well, if you look at what actually happened, at least from testing that was done on campus, which does not give us a complete picture of the outbreak, but might be a reasonable proxy for symptomatic cases, we actually see a peak about 90 days in of about 200 active cases. Now, our model's not totally right. I don't believe that by any stretch of the imagination, but what we seem to see is that we can find a reasonable set of parameter values to actually model what actually occurred, which you can then use to study lots of other things. So I'll sort of stop there and allow time for any questions.
Thank you, Quinn. This is super fascinating. Mathematical modeling is relevant. And when I think about all those math classes I took that I did not enjoy, I'm like, well, there is cool application and you're proving it. So any questions from the audience? We do have one in the chat already. And I think it was about that first image that you shared, the sort of ink blot splotch, where you showed the different colors. And you mentioned that the grad students are kind of outliers. So is there a reason that they're so far away? Is it mostly because they live off of campus that they have fewer opportunities for connection?
So that's certainly part of it, although many of our undergraduate students also live off campus. So that alone is not enough to explain it. It tends to be, most of the structure of this graph, I think, can be explained by curricular requirements. In fact, many graduate programs limit the number of dual listed courses that you can take. So the opportunities for an undergrad or for a graduate student to be in a class with an undergraduate student are actually sort of structurally limited by the curriculum itself. So I think that most of the structure of this is due to curricular requirements. The same reason freshmen are connected to so many people. They're all taking gen ed classes, and gen ed classes sort of don't respect your major in that. I don't mean respect in some sort of, this is not a diatribe against gen ed, but it does create this sort of artificial mixing where when you look at juniors and seniors, they tend to be a little more isolated to the same group of people. Now they're having more contacts with that smaller group of people, but overall their total number of contacts is smaller. And so, sorry, I'm just seeing there's another one here. Do you believe contact tracing was actually helpful in minimizing case counts? I think that's outside the ability of my expertise in this model to say. And so, you can try to build in something like contact tracing. As with many things, it's sort of behavioral in some sense. I mean, the ability to effectively contact trace is limited by a number of things, both sort of political and structural and all sorts of things. And so I don't have an informed opinion on that, because I think it's outside the scope of my model to say such things, to really answer that question. But we have excellent public health folks who might have a good opinion on that, a good factually based opinion.
Michael, you unmuted. Do you have a question?
Yeah, I had a question, a follow-up question to the questions that have been asked. Can this model, or based on how you model it, do you think classification of the students, freshmen or seniors, would fall at different categories, or all the students were the same?
So one thing that I've actually been working on doing recently is trying to understand sort of, I don't know that the best way to classify them is, or to really understand like clusters, is to think of it in this term. So mathematically, there's some things you can do to mathematically identify clusters out of this network. And so one of the sort of open things still to do is to work and try to do that, is to understand what are these clusters, both mathematically. I mean, there's mathematics just to identify the clusters. And then once you do that, the secondary question is, does this cluster mean anything in practice? I mean, mathematically, you can say these students form a cluster, but if there's no sort of coherent way of identifying and contacting or doing anything with those students, then it's an interesting mathematical exercise, but practically speaking, it doesn't do you any necessary benefit. You know, one of the slight exceptions to that is many people have discussed sort of the idea of pooled testing. So the idea is you don't need to test all the students, but you need to just test sort of the right sample of the students to get an idea of what's happening. One really smart way to do that, in my view, is to mathematically identify the clusters, randomly sample the clusters, and then I think you actually get a truer sense with fewer tests of the actual COVID situation on campus. So that's really, I think, where the clustering and understanding the clusters can make sort of the most difference.
Will, you have a question?
Thank
Yeah,
you.
Quinn, first of all, from a university housing standpoint, when I asked the question, what in the world is going on? Thank you. This explains a lot. Appreciate you. Second, in this model that's on the screen, I know you talked about the green, blue, red, or orange. I may have missed this. What does the yellow transitions in between, what do they mean?
So it's let's see if I get these right. Red or freshmen,
Okay.
orange, which you can sort of maybe kind of see in here, are sophomores. Actually, sorry, yellow are juniors, green are seniors, and blue are graduate students.
Ah, okay. Okay.
Sorry, I should have put a legend up here. That would make this a lot more readable.
No, okay.
But yeah, so part of what I didn't say is that the way this is generated is you're actually sort of imagining that each of these two things, if they're connected by a spring. So people who are connected to a lot of other people end up in this cluster in the middle. And people who are maybe only connected to one or two, well, they're only connected by one spring, so they can sort of get pretty far out here. The other sort of slight detail I left out is that I've actually remade, this is actually includes faculty members also. They're hard to distinguish because they're black, but I'll give you, so here is one faculty member. Notice that faculty member, this is a faculty member who taught undergraduate and graduate courses. That one faculty member provides a possible transmission link between two different groups. So, you know, one other thing we sort of discussed when we were talking about all of this was, you know, you could actually identify some possible ways of disconnecting this network, right, to prevent possible transmissions. If you reassigned this faculty member not to teach that graduate course, which carries all sorts of implications, and I know that faculty member doesn't want to hear it's because Quinn's model said so. But if you do that, you remove that transmission link. So again, and I'm not claiming this is the best thing to do, but it's another data point in trying to solve a very complex problem.
Awesome. Thank you so much for your presentation, Quinn, and food for thought. It's so super relevant. And again, thank you so much. We'll go on next to Dr. Mai, so you can share your screen and we will carry on. Keep the good questions coming. Quinn, I know you have other commitments today, so we will not be offended if you need to hop off.
All right. Thanks so much. You all have a great day.
Thanks, Quinn.
So I'll get started.
Yes, please.
Can you see my screen?
Yep, we sure can.
Great. So I teach in the Department of Philosophy and Religion. My teaching specialty is Asian religions and comparative religion. I focus on religious approaches to death and dying, the history of the afterlife, and mourning rituals in various religious traditions. This particular research has been made possible by the generous funding of grants from the Board of Trustees, the URC, and the College of Arts and Sciences summer research grants. My research specialty requires that I consult medieval texts, and while interlibrary loan is very important for me, they can only do so much because sometimes the materials I need, libraries just don't have. So this is why for this project, I had to travel and visit archival libraries in Hanoi, Vietnam, and Taipei, Taiwan. For example, here's a short list of texts which I use in my research. Each text listed, which I don't want to go into deeply today, has various versions and editions, some typeface and some manuscript, handwritten editions. The standard scholarly practice requires critical examination across different editions. So it's very time consuming and requires direct access to archival materials. Some of the religious themes I particularly focus on are, for example, some of the stories that I find in these texts are stories about the lives and deeds of gods and goddesses, the apotheosis of warriors, chaste wives, and loyal officials is another common theme as well. Often you'll find ghost stories, specifically stories about hauntings of restless ghosts. I've written an article on this already. Another fascinating topic is return from death narratives, people dying, going down into the underworld, visiting previously deceased members of their family. And for this particular project, I'm looking at stories about love affairs between humans and ghosts, demons, goddesses, and immortals and such. So what is the importance of this material? This type of material gives us insight into the religious imagination of pre-modern culture. The texts that I look at shed light on the practice of Buddhism, Taoism, Confucianism, and popular religion in traditional pre-modern Vietnam. The central question for this particular project is the karmic discourse used to talk about love best described as Buddhist, Taoist, Confucian, or a part of popular religion? The short answer is yes, all of these. For example, in particular, the paper I've just published, I did a close reading of 4 stories and provided translations as well. And I analyzed the ways in which love is imagined as constituted by karmic bonds over lifetimes. I look at Taoist uses of the concept and Confucian uses. Taoists tend to frame the bond of love as a possible means of attaining transcendence from this world, whereas Confucians tend to use the trope of love as a way of reiterating the importance of the family and the importance of the inheritance of fathers. So basically it tends to emphasize the patriarchal framework. When I think about this topic, there are also other comparative possibilities as well when we think of the role of religion and love in, for example, Christian mysticism, Islamic mysticism, Hindu devotional worship, and so forth. So I'm sorry, but due to time constraints, I don't want to get too deeply into that here, but if you're interested, I'd be happy to send you a PDF copy of the recent article published in the Journal of Vietnamese Studies. Thank you very much for your attention.
Thank you so much. I feel like you just gave us this tiny little peek behind the curtain, and I have so many questions. So I will let someone else start with the questions, and we have a little bit of time. We have actually plenty of time for questions.
Okay, great.
I have one that you mentioned, you just mentioned patriarchal influence, and I was wondering if there was any evidence of matriarchal influence in any of them.
That's a good question. There really isn't, because the type of texts that I look at are written mostly by classically highly educated men. And first of all, the viewpoint that you'll get is going to be from their viewpoint. And so you're not going to find that. And historically also, we do know that in pre-modern times, there wasn't much of matriarchy as a social phenomenon. If it did exist, it probably is fictional, in a sense of some counter narrative about what is predominant. Thank you for that question.
Thank you for your presentation.
So can you talk a little bit about how you identify the texts that you want to do close readings of? So are these like historically well-known texts that people of the region would talk about and know, or have you identified them through other archival research and knowing that these are the texts that you need to go and comb through? How do you identify the texts that you're going to do close readings of?
Right, so that's a really good question. Usually there is a collection of compilations of texts that are well-known already, but the particular texts that I look at are not well-known, and this is why it's really important to go into the archives, because there has been no published research on it. And so there's a lot that we just don't even know, and we don't even know what types of questions to ask, because no one has gone into the archives and just gone through the different manuscripts that are available.
Yeah, that's fascinating. And I assume that's because you want to preserve cultural history and public knowledge of this type of cultural history.
Correct, right. So religious studies, we try to use as many different types of material evidence as possible, from text, manuscripts, to art, to images, icons, in order to try to interpret what people felt, thought, and talked about in terms of the religious reality that they lived in. And because it's, the farther you go back, just the less material you have. So you have to be really open-minded and try to use different types of texts in order to derive different reconstructions of the past, what people thought about reality, basically.
Fascinating. And so there's a question in the chat about how does Confucianism play into a culture's view of life after death? And Benjamin is asking because his fiance is Korean, and she describes Confucianism as ingrained in their holiday where they honor the dead.
Oh, right. That's very interesting. So Confucianism is really important in the cultures of East Asia, which includes China, Japan, Korea, and Vietnam. And it plays a central role in emphasizing the importance of the family, and the family as the unit of identification. So typically, to overgeneralize, the individual is always understood in relationship to the group, such that one way that this is reiterated is precisely through religious rituals such as ancestor veneration, sometimes called ancestor worship. So the ancestor altar then becomes a way of interacting with the dead who went before, the lineage of ancestors, but also it's a way of imagining oneself as connected to the past, but also connected to the future, because it's your children who watch how you engage with the ancestors, who then mimic how you behave towards your predecessors. And then it reiterates that whole ideal of family harmony, which is central to East Asian culture.
Thank you so much. Any other questions? We still have another minute or two.
I have a question. It's more broad-based. How long does it take a religion to change? Because I think the way religions are right now, it is different from the way they were probably 30 or 40 years ago.
Right, that's a great question. That's going to take me about several hours to really unpack, but there are many factors in which religions change, right? So we can think of society, economics, politics, material culture, books, literature. So you have to kind of account for all of that. And one of the main reasons in my mind that is changing so fast is because of social media and the internet and the quick pace of communication. So really is proving a big challenge for people who study contemporary religion.
I imagine it's just like accelerated on some sort of spectrum, right? It's
Right.
sort of held traditional for a long time. And yeah, it's super interesting.
Thank you.
Thank you.
All right, any other questions before we move to our final speakers? All right, awesome. Thank you so much for sharing and your presentation. And I did not ask the speakers of this because we'll have the recordings, but I would really like copies of all of your slides so that I can, so we can have them to share for folks and review, if that would be easy for you all to share. All right, Will, I'm going to turn this over to you and Brandy, and they are our final dyad of speakers for this lightning talk. So y'all can take it away and share your screen.
All right.
Yeah, before I jump in, I was going to say we're really happy that we've got some biology, some philosophy of religion, some math, some other sciences folks here. In my role as Director of Inclusive Excellence, I get to look at a lot of our student data. And so as we're moving into this presentation, it's important for us to remember that when we look at some of the student data, we have some pretty high DFW rates. So that's, students who either earn a D, an F, or withdraw from certain classes, and that impacts retention in the discipline in the field. And we know that some of those highest DFW rates for our students of color, and particularly some of our African-American students and African-American males, are actually in the sciences, math, and philosophy and religion. So we hope that this actually can contribute to our co-presenters as well today.
Okay, we'll go ahead and jump in. And first I'll say thank you for the research offices for giving us the opportunity to do this research and getting the chance to take a van across North Carolina to do the research and coming back and presenting our findings and having some practical solutions from this. So what we did was we traveled around, like I said, North Carolina, and we did focus groups to examine the skills, habits, characteristics, relationships to learning for 26 high achieving Black males in their first year at HBCUs. We wanted to see what were the things that they have that when they came in, they brought with them for success and the things they learned while they were in that first year. So this came at a very important time as we were wanted to get ahead of that narrative that, oh, Black men are failing. Yes, we see the data on that, but how can we reverse that to make it that we see that they are succeeding as well? And so we did this to elevate, and then our opportunity to make an article out of this as well.
So in terms of defining Black male brilliance and how we're talking about this, and I linked, I gave you all a link to the PDF copy of the article. After we did all of the interviews and of course, sifted, sorted through our data through various analysis processes, we recognize that these guys bring, these high achieving Black males bring these common academic success characteristics. So we know that they have high levels of academic ethic, or what some might call pro-school values. That includes motivation, persistence. They talked about examples of how they continue to persist, of sacrifice, of discipline in some of their habits. But why we called this Black male brilliance, again, encountering that narrative that we often hear that's located in a place of deficit about Black males and their relationship to learning. All of these pro-school, high-level academic ethic characteristics really combined with these significant barriers. So persistence through damaging racial stereotypes and racism, these structural barriers that exist, while also maintaining a strong and positive sense of identity as Black men. So we break this down more in the article itself that we can't really do in this lightning round talk, but this was really how we were defining Black male brilliance. And some of these definitions came directly from those focus groups and the data from the guys.
Yeah, so here are some of the findings that we found as we interviewed from the focus groups. One was disproving stereotypes or proving people wrong. Like, I want the power to prove people I can do this. So one of the participants said this: it makes me feel accomplished when I exceed the expectation people have already set for me, even though they don't know a single thing about me. So they want to prove things wrong for those biases that people have. And so they thought, well, he's coming in, he's going to need help and so forth, but they don't know the barriers that the student came from, or what his motivation is, or what he wants to do in the future. So with that said, he wants to prove everybody wrong that doubted him going forward for success. Second was strength in their identities as Black men. It's inspiring to see people that look like me and be able to do great things. So they want to make sure they see others that they can see, support, have a peer model to help each other and lift each other up to see, wow, we're both succeeding at the things that we want to do. It may not be the same thing, but we at least can be in the same circle to push each other to go forward. Speaking of forward, second was forward thinking, forward aspirations. It was huge. They had so many cool aspirations to see, I want to be this, or I want to be a doctor, or I want to be something in the academics and not just sports related. It was good to see that they wanted to be something on an academic level that was challenging, and they had huge aspirations to help their community, their family, and themselves, and just be a role model for success overall. And then we're looking at the cognizance of limitations. So these are the things that they said, these are the challenges we have to face. Number one, societal challenges. We know there's a lot of challenges going on in our world right now, from political to socioeconomic to race-related. So there's a lot of challenges our Black males had to face, not even just coming to college, but in their neighborhoods going forward. Next, personal challenges. They have their own internal struggles that they must heal from and have to get support from that. Third, as we had talked about, that math. Math was one of those areas that needed a lot of support. And if they didn't feel or have the motivation to go get tutoring or help, or like, I don't want to seem as a failure for going to tutoring, well, then they're going to fail. So we had to get them to overcome that and talk about that. Then time constraints. It's different from coming from high school to college. How does that look to transfer those skills from high school that you may or may not have, and then actually learn the skills you need to be ready for the time constraints that are going to be in college? And then finally, trusting others. That was a huge step in seeing, well, can I trust this person? They're going to have my best interests at heart. Are they going to be able to support me, role model for me? And when I need them, are they going to be there? That was the biggest thing. So that, are you going to let me down? So they wanted to see people that were ready to support them right off the bat and be there for them.
Thanks, Will. So moving into a couple of the implications and recommendations that we found from this study. Number one, for us as educators, whether we're faculty or staff or whatever our role is making sure that we're checking our biases and assumptions about our Black male students. We have so many implicit biases. We all do. We all carry implicit biases. And sometimes those can come out in our behaviors, in our comments, and in our interactions with our Black male students in a way that we don't even realize. So doing some of that personal work that's related to our professional development work, to be positive educators and inclusive and equitable educators. Integrating positive representations of Black people and Black men. That quote, we want to see people who look like us do great things. They want to see that in the classroom. They want to talk about that. And I thought about this as Dr. Mai was talking and presenting, even just saying something, if you're in a field where there really aren't a lot of Black people or Black males represented because of the history of your field, just being able to say that and talk about that is actually something in the research that says supports a more inclusive environment by acknowledging that history and that problematic exclusion of Black people and Black males from your field or your discipline. The next one was create strong programs that draw from Black male students' assets and their cultural community wealth. So really thinking about how to draw on those strengths. And then one came directly from a quote from one of our participants. He said, "Educators, please have faith in our ability to succeed and help us believe in our brilliance. We need to know that you believe in us, and we want you to help us believe in ourselves." And then intentionally build relationships with Black male students. These Black men made a number of specific recommendations of what those relationships should look like. And we're going to detail those more specifically in our longer session from 3:30 to 4 o'clock today. But since we do have just a couple, an extra minute, I actually threw the slide in from that presentation into this presentation, just one slide that I think would be really helpful here. And so when they talk specifically about their recommendations for educators, they said, be consistent, committed, and invested in our future. Develop intentional, meaningful relationships with us. Consistent care and commitment. They need consistent care and commitment. And this is what it looks like. It looks like supporting them in good times, hard times, imparting life lessons, and actually being intentional with regular check-ins with them. They said, show us some love and be patient with how we respond. One of their limitations that they were cognizant of was trusting people. So if at first you don't succeed, try again, is what came up in one of our focus groups. We may not respond to you positively right away. Keep on showing us that you care, and eventually we will respond. But we need to know we can trust you first. And then again, have faith in our ability to succeed and help us believe in our brilliance. So that was some of the things that they detailed in terms of what does it look like in terms of supporting them from the perspective of an educator. And then the last piece here is that institutions really need strong programs that draw from Black male students' assets and their cultural community wealth. That was actually on our previous slide as well. Will, did you want to provide us with any closing comments or connect to the BMEI?
Yeah, we did start our residential learning community to talk about, to respond to some of these implications. And so we do have a first year initiative on campus that staff is myself, Dr. Bryson, and others have been committed to. And so far it's going great. And we're hoping to get more on board to helping these students of color and Black males with making sure their overall success is better. So thank you. And now we'll open it up for questions.
Yes, thank you so much for this presentation. And as they mentioned, they've got a longer session coming up at 3:30. So if you have a half hour, I would hop on. It's got some good practical advice, it sounds like. And so I'm looking forward to hearing that session as well. So we have a few minutes before we wrap up for questions for them. I have a question to start, and I just have the advantage of knowing a little bit about this initiative and this project. So, Will, I know that you guys did this research at HBCUs in North Carolina, but you mentioned that we have a residential community here on our campus. So could you talk a little bit about the application from the stuff you've learned at HBCUs and how you're applying it to a primarily white institution like App?
Yes, so after our research with the HBCUs, Dr. Bryson and I was thinking, what does this look like at PWI? So we did do a comparative study, And we did some focus groups with students here at App State, compared them to HBCU studies, and looked at what the significance of what was needed at PWIs and at HBCUs. And we saw that there were some differences, but actually there were some similarities of the things they needed, like trusting, needing that support, and so forth. And so that study, from seeing those two studies, that's kind of where the initiative was born out of, and seeing that there was others on campus that had the passion to want to do that as well.
Awesome. Thank you. Michael, you unmuted. Did you have a question?
Yes, I had a question. Thanks, Will and Brandy, for that good study. And it reminds some of us some of the things we think about or we have talked to people about. So I just wanted to ask if you ask them, or in your study, if you have a way of knowing how they did with imposter syndrome.
Ooh, great question. And Brandy, you may can help with this as well.
Yeah. You want me to go ahead, or?
Sure, go ahead.
Okay. I was going to say, I think that some of them did struggle with imposter syndrome, but many of them did not. And particularly with our guys at the HBCU. Now, I will say the pandemic really messed us up in terms of we were going to do this more robust comparative study at App State, but we only got a handful of participants. So that study was that part of the study wasn't super robust. But I would say that the Black males attending App State and the Black males attending PWIs, in terms of what we see in the research literature, struggle more with imposter syndrome than the Black males who attend HBCUs. So I think that speaks to our call and our need as educators at a PWI to really step up and think about ways of supporting them. Great question.
Did a follow-up question is a little bit different, but did they compare, are there other studies that have shown some of the, I know the other races will also be going through some of the things that they mentioned. Are there studies that have shown the effort comparing the Black males versus other races in the effort they put in to fight those stereotypes or things that they have to overcome?
Yeah, I'm thinking about the term racial battle fatigue. Did you want to speak on that or something else, Will?
No, I think you're spot on. Go for it.
Yeah, so the literature around thinking about racial equity in higher education, particularly in PWIs, there's some authors and scholars who write a lot about racial battle fatigue and the amount of mental and emotional labor that is required that pulls us away from some of those other cognitive academic learning parts of our brain. And particularly when that racial battle fatigue is combined with trauma, which really, puts us in the back part of our brain, the survival brain when we deal with trauma, whether it's racialized trauma or other kinds of trauma, it really pulls us out of that prefrontal cortex and into more of that unconscious thinking brain, survival brain that then impacts learning.
Thank you.
Yeah, we saw a lot of fight or flight, seeing how can I just survive being here and where I'm at, and thinking about, well, if I'm hearing I'm supposed to fail, so am I supposed to fail? So like I said, we wanted to flip that and see, well, what are the successes that you need so they can start ingraining that in their brains going forward.
Thanks.
That reminds me of stereotype threat. And how when you find yourself, you see you hear about a stereotype and you're like, oh, I'm that stereotype. Is this going to happen to me? And so it impacts your your performance. I think that's super interesting. And I hate to stop us, but we're at 3:02. So thank you everyone for joining us for this lightning talk. And please be reminded, do check us out. We still have 2 more days of events and sessions, plus 1 more today. So if you liked how we ended things today, come back for more of Will and Brandy at 3:30, back in the same spot. So we'll see you all back here. Thank you all so much for joining us, and we hope to see you at some future sessions. Bye, everyone.
Take care, everybody.